Before I jump into the substance of the scientific presentation, I just wanted to say a few words about Jim Simons, because I had the privilege actually to get to know him a little bit, and it was a wonderful experience for me. It started with his email in 2012, very terse and to the point, here's an invitation, I hope you can join us. Sincerely, Jim Simons. And attached to the email was this letter, which had me at Berkeley, not Stanford, and I figured this was written by his assistant or something and sent out under his name. And I replied, oh, I'm already scheduled for something then, I'll see if I can rearrange. And then I promptly forgot about it. But Jim did not, because a week and a half later, I got another email, and it was clearly from him. And he said, except for you and Corey Bargman, everybody's agreed to come to this meeting. And by the way, I'll be out in California the day before, and you can fly back with me on my plane. So I rearranged everything and said, yes, I'd be happy to come to the meeting. And this was the Buttermilk Falls meeting that marked, Jim moving from Renaissance to spending full time with the foundation. And it was a wonderful meeting. We had an amazing conversation on the trip there. The meeting itself was terrific. And then afterwards, Jim wrote a very personal note, which I was grateful for. I just couldn't imagine someone with that many things on his plate spending the time to do that. And it was very meaningful. And he always was willing to make time to chat whenever I was coming to New York, and I was very grateful for that. He was a very special person. And if you haven't read the biography, it's very interesting. He was such an original person in so many ways. And if you look at some of the quotes in there from him, and think about them in the context of the world we're living in now, where we're going through this revolution in AI, and people are obsessed with getting data to make predictions, and building lab-in-the-loop systems that'll do science autonomously without people interfering. He was way, way ahead of all that. And I'll return to some of these ideas in the second half of my talk. So let's start sort of at the very beginning and get everyone on the same page. Cells are sort of the fundamental quanta of biology. They're the smallest indivisible units of life. This is a beautiful picture painting by the artist and biologist David Goodsell of what he imagines the last universal common ancestor to be. And it shows the cell dividing, and he's taken sort of all the structural knowledge we have about the constituent molecules of a cell and tried to put them in place in what he imagines the very dense, packed interior of the cell is, with all of the protein machines and enzymes. The long stringy thing is the genome or the DNA, and you can see the cell wall and the lipids and the polysaccharides and so forth. And so the cell is a very, very complex unit. And the genome inside it is made up of a chemical called DNA. It's the molecule of genetic inheritance. It's got this famous double helical structure, and information is encoded in the DNA through the chemical alphabet. We'll just call it ATCG for this talk, but the famous base pairing is the way information is written in there, and that's the template for making the proteins and the protein machines. And the story of the structure, the discovery of the structure of DNA is a sort of very interesting classic one in the history of science. The three key players were Jim Watson, Francis Crick, and Rosalind Franklin. And you can read some of that adventure in their own words. In the case of Watson and Crick, Franklin tragically died very young, but there's a beautiful biography of her called The Dark Lady of DNA. And it was a very complicated story about the history of science, and it's worth understanding. They called it the secret of life. Watson went around telling everybody, we solved the secret of life. We started getting the structure of DNA. But there's much more to it than that, as we'll get into in this talk. In trying to understand how that information in the DNA got put into use in the cell, Crick created something called the central dogma. When trying to understand information flow from the DNA to the proteins, the idea is it's copied from DNA into an intermediary molecule called RNA. That's then translated into the protein. And DNA gets copied when the cell replicates. And what we learned much later is that sometimes information can be copied back from RNA to the DNA through the action of certain viruses. But for the purposes of understanding how the cell works, this is sort of kind of the basic construct. And it turns out, sort of long story short, the proteins tend to be very hard to measure. The DNA is the same in every cell in your body, so it doesn't explain multicellularity to really sequence that. But that RNA intermediate tells you which genes are turned on in which cells, and it's fairly easy to measure. And that's going to be the story tonight of how we use that to try to tease apart some of the secrets of multicellularity. And this was all discovered in the context of a model organism, E. coli, famous bacterium, and it sort of underlies an enormous amount of biology and kind of the universality of that. Monod tried to capture in this phrase, anything true of E. coli must also be true in elephants. Not entirely true, but it captures sort of the spirit of using a model organism. And if you're interested in all the history of these written by a third person, there's the classic history is, and this will be my last book recommendation tonight, Eighth Day of Creation by Horace Freeland Judson, which goes through not just the discovery of the DNA structure, but the discovery of how information is processed, how genes are regulated, and then how protein structures are determined. Okay, let's talk about DNA as a molecule for a moment, because it's astonishing when you think about it. Every cell in your body has 2 meters of DNA in it. And your cells are not very large, just a few tens of microns. Every one has 2 meters of this molecule. If you took your chromosomes, laid them out end to end, that's how far it would go. You have roughly 30 trillion cells in your body. So if you took all the DNA in your body and laid it out end to end, it would go from the Earth to the sun and back more than 200 times. How does that work? And how do you fit all that into your body? The reason is that DNA is macroscopic only in one dimension, but it's atomic in the other 2. And it's got a diameter of only 2 nanometers, and it's packaged up and wrapped up, and that's how it all fits. But it's sort of this amazing thing that is both macroscopic and atomic at the same time. And that makes it a very interesting tool for experiments. You can see it with your eyes, and yet it behaves like a molecule. And early in my career, that's sort of what I was fascinated with, trying to image DNA molecules, study its physical properties as a polymer. This is a movie we made when I was a young professor, taking DNA using optical tweezers to tie it into a knot by making it cross over. And as the knot gets pulled tight, you can see it kind of contracts. The bounding motion of the molecule is evident here. This is in a very viscous solution, but this is was just a source of unending fascination to me to be able to watch molecules and measure forces and look at their dynamics. So that's it as a physical molecule. But it's also, as I mentioned, a place where information is stored. And over the last many decades, the scientific community has figured out more and more powerful ways to read that information out, the ATCG sequences of any given genome. And I got involved in this from the perspective of using these single molecule techniques to make sequencers, and that marked a sort of pivot in my own research career from biophysics to genetics and genomics. And this sort of summarizes how fast the field is moving in the early 2000s. The cost of sequencing a genome, or megabase pair, going down exponentially over time, faster than Moore's law, and the number of genomes increasing with time. And so we got to be very good at sequencing genomes of many different creatures, the human genome, many different individuals. I sequenced my genome to try to understand what was there. And that put the field to sort of an interesting point, which kind of led to the title of the talk. How does one genome yield many different cell types? And people, speaking loosely, used to say that the genome is the blueprint of the organism, but not really. It's more of a parts list, because every cell has the same body. If they have the same genome, if they have the same blueprint, they wouldn't be different. They choose very different identities. A neuron, very different than a lung cell, very different than a muscle. So each cell type is picking different parts from the genome and only choosing to express those parts and to make different identities and functions. And thus far, it has been impossible to predict the various cell types of an organism from the genome alone. And famous biologist Sidney Brenner pointed this out in his Nobel Prize address, which was super thoughtful. He was a great wit and loved to poke people, and he wanted to poke the genome people. The human genome had just been sequenced in, I don't know, it was 2002 or so. And he pointed out the job is far from done. And knowing the genome is very different from understanding the biology of the organism. And this was his example. He also had this great line, biology is drowning in a sea of data and starving for knowledge. And I think something to remember in today's world where people think data is the answer. It's not really. It's just a route to getting knowledge, and you can't, doesn't end with getting data. And so he suggested 20-odd years ago this program of trying to work out all the different cell types, a molecular definition of them, and said, oh, if we worked really hard on this, the scientific community can get it done in 20 years. And more or less that has happened. And it took a long time. As I mentioned, the route to getting there was through measuring RNA and doing it in single cells. And that's become a very hot field in recent years, but it was many decades in development. And so really 30 years of technology development to get us where we are now, where it all works. And it goes back to 1990. So when PCR was invented, it was the first biochemical amplifier, almost from, that was late 80s. And right from that point, people began thinking about different ways to amplify nucleic acids. And in 1990, Eberwein and Isco both published papers in the same year showing that it was possible to amplify the transcriptome of single cells and to make single cell cDNA libraries. However, they had no way to read it out. And so they were probing with southern blots. You can Google what that is if you're a young person. And so they could look at one or two genes and understand that maybe they had amplified the transcriptome, but they didn't really have access to the full complexity of all the RNA from a single cell. And it took another decade or so to get that done. And in 2003, there were 4 papers published by these folks who used hybridization microarrays to read out large numbers of genes from single cell cDNA libraries, both homemade and commercial. And at that point, they were able to look at upwards of 10,000 genes per cell, and so could really kind of understand the complexity of what was going on there in terms of the gene expression. And then inevitably, it shifted to sequencing instead of hybridization to read out the information. And initially by Sanger sequencing by Morose and Kandel, and then on to next generation sequencing, which is how we do it now. And so this all took a long time to develop. And I think the only piece that wasn't solved by all this was the ability to do it at scale. These were all bespoke experiments with a handful of cells. It took a lot of work to do it, and there was no way to really do lots and lots of cells. And that's how I got involved in the field. My group had been working on microfluidic automation. We'd built little chips with valves and pumps and things like that. And those we were exploring as tools to automate biology. So we did that. A company I started called Fluidigm made the first commercial device for doing single cell genomics. And the chips had the amazing throughput of 100 cells per chip, which was just phenomenal. And any lab could do it. Hundreds of labs were using this. It really got the field launched so that any biologist could start dissecting their system this way, their organism of interest. And then the sort of desire for more cells, the hunger for that grew and grew. And a next generation of technologies, also based on microfluidics in my lab, on water and oil emulsions, was developed, this time by a group in Boston, a number of groups in Boston. Goes by names like InDrop, DropSeq, so forth. That became the second generation. It lets you do thousands of cells per experiment. And in working that out, we sort of realized it was now possible to do this at scale, to really dissect any given tissue. And the experiments sort of went like this. You take the tissue of interest, you would chemically, enzymatically dissociate it, and then use one of these tools to do the single cell encapsulation and creation of the cDNA libraries. And every cell had also a DNA barcode attached to it, so you could read out which cell it came from. Everything is then mixed together, sequenced, and then you compute on this data. And so every cell was essentially giving you 20,000 dimensions of information. It's represented by a 20,000 dimensional vector. There's roughly 20,000 genes in the human genome, and every dimension is a different gene, and the number in that dimension is the number of mRNA transcripts from that cell, telling you whether that gene was expressed or not, and if so, how strongly was it expressed. And so there's many interesting things one had to do on the computational side to understand that, because we as humans are not very good at thinking in 20,000 dimensions. And there was great computational tools to do that and do dimensional reduction, clustering, and so forth. So with all that in hand, we realized that as a community, there was an opportunity to do some really large projects and to try to build cell atlases of entire organisms that all the pieces were in place. And that's sort of how I got involved in starting the BioHub. And for me, the motivation was I wanted to do these large team science projects to build cell atlases that would be phenotypic companions to the genome and help us understand the molecular definition of all the cell types. And so we did that. And sure enough, as Brenner predicted, it was almost 20 years to the year after he said it could be possible. At the BioHub, we focused on doing whole organism cell atlases, we were getting 18 and then 24 tissues per individual donor, and trying to build comprehensive atlases. Did it for mouse, fly, lemur, human. I'm going to sort of just mention a few highlights of some of the mouse work, but spend most of the talk on the human data and then how we've been thinking about it going forward. And these were, as I mentioned, sort of team science projects. Each paper had 150 authors. We had collaborations with groups across all the Bay Area institutions, and it was just a ton of fun to do, and a very different kind of science than anything I had done in my career previously. We started with mouse. Tony Whisker-Ray, who's a colleague at Stanford, was in my office saying, why don't we look at transcriptomes of aging mice. He was thinking of doing bulk tissues. I said, well, we've just started this institute. We should do single cell atlases, not just of the mouse, but of the mouse as a function of age. And so we did that. And one of the challenges was the oldest mice, there weren't very many of them for obvious reasons. The 30-month-old mouse is equivalent to a 100-year-old human. So we knew there wouldn't be many, and so that's what forced us to develop this whole teamwork method of working together, of getting 18 tissues from the same animal. And that proved key to everything we did later in all the other organisms, including human. And Spiros Dermanis and Angela Pisco were two young people, the group leaders of the BioHub, who helped drive all that. And we studied sort of natural effects of aging on the transcriptome. I'm not going to get into the details, that's all published. We also tried to reverse aging. So folks in the aging field, Tony and his colleagues, had figured out that if you suture together two mice, so they have a shared blood supply, that's called parabiosis, and you have a young mouse and old mouse sharing the same blood supply, the old mouse becomes younger, and the young mouse has accelerated aging by certain physiological measurements. There's a whole community of people studying this. And so we decided to make atlases of those mice as well. And I was 100% sure we were going to completely debunk this field. I didn't think any of it was real. But it turned out I became convinced there actually is some interesting real biology going on at the level of transcriptomes through parabiosis. And again, that story you're welcome to dive into. So let's turn to human, which will be the subject of the rest of the talk. From what we learned at mouse, we decided to take the same approach to human and get many different tissues from the same donor so that we could hold many things constant, hold the genetic background constant, age, epigenetic effects, environmental exposure, and be able to do comparisons of shared cell types within the same different tissues from the same donor. And initially, the first atlas was about 500,000 cells. Bob Jones was the person who quarterbacked it all, a senior research person in my lab, but it included many people. I won't go through all the names here, but these are some of the pictures. You might recognize some of the folks. We not only did the transcriptomic cell atlas, we also had pathological images of many of the tissues and did microbiome analysis on a subset of them. This is sort of what it looked like. Here we go. So each column is a different human donor. Each row is a different tissue. We did upwards of 24 tissues per donor. And everything to the left of the dotted line was the first 500,000 cells in the first paper. Everything to the right is the next 500,000, which has not yet been published, but I'll tell you how we're analyzing that. And you can see over time we got to be very, very good at getting many tissues from the same donor. That came about through a terrific partnership with an organization called Donor Network West. And they are a nonprofit organ procurement organization. They're the ones who help people who need organ transplants and match donors with recipients. This particular one serves Northern California and Nevada. And for organs that aren't transplanted, they can be used for research with the consent of the families. They were just terrific partners in helping us find donors and families willing to participate in this research. It was by far the most complex thing they've ever done. We worked very, very closely to figure out how to do this. Typically what would happen is they would give us a call. We have a donor coming up. We're getting ready for a transplant in a few days. Is this someone that would be suitable for your study? We'd look at the medical records, make a decision. And if so, they would then get the family's consent and if possible, if they were willing, and then we'd go and do it. And it generally always happened at night for some reason. These were 24 to 36 hours of continuous work. The surgeons would operate, taking the tissues out one by one. They'd be couriered over to the labs where they were dissociated and then converted into cDNA libraries to be ready to sequence. And it just required an enormous amount of dedication from the staff and the students and the postdocs. My role in all this was basically to deliver pizza in the middle of the night, and they did all the real work. But this is sort of an example. And you can see they're all wearing masks. This happened during COVID times, which made it another layer of complexity. But really, just an amazing amount of dedication from these folks and the many others whose names are here. As I mentioned, it was a very, very large collaboration. So let's turn to some of the things we've learned from these data sets. So I mentioned the first secret of life. The second secret of life, which Jacob and Monod discovered, was that some genes control other genes, also known as regulation, okay? They again discovered this in bacteria, but we now know it's a universal principle. And the way it works is that proteins will bind to the genome and control whether or not a gene is copied into RNA. And that's why not every gene is turned on in every cell. There's proteins that will block that, others that cause it to happen. The ones we'll focus on here, transcription factors, are the ones that sort of control gene expression. And when they bind, they'll help recruit the RNA polymerase, which is the enzyme that copies the RNA from the DNA. So that's how gene regulation works. Now, in the human genome, there are roughly 1,630 so transcription factors that have been predicted from the genome. Some of these have been studied in great detail, are very well known. Others are just hypothetical transcription factors that have been based on sequence similarity, and people have predicted their existence, but nobody knows the biology. So we decided to take that whole list and ask if we could see which were being expressed in our Tabula Sapiens cell atlas. And the answer was all of them. So we now, for the first time, have identified the cell type specific expression of every transcription factor in the human genome. And here's an example of that data. So here, every column is a different transcription factor, every row is a different cell type, and the blue is the intensity of expression. And many transcription factors are expressed in only a handful of cell types. We computed something called tau, which is a statistic for how specific the expression is across cell types. These are generally the low tau ones here. And if you look at well-studied cell types, all the usual suspects are showing up and makes sense with existing knowledge. But basically, there's a whole bunch of unstudied ones, both at the level of transcription factor and cell type, where we now can work that out. We know exactly which cell type to study to understand these transcription factors and where they're used. So it's like a whole recipe book. And if you think about how cell type reprogramming is done, this kind of becomes a way to do that all in one fell swoop for all the cell types you can imagine. Whereas other transcription factors at the low end of the tau statistic are expressed very broadly across all cell types. And this shows sort of the lowest tau. And those turn out to be very interesting as well. Those are transcription factors that are ubiquitously expressed by every cell type, and they're responsible for functions that every cell has to do, like stress response, proliferation, metabolism. And you'd expect all of these to be very well known, right? And every cell does it. But it turns out that on this list, we found many well-known ones, but there's also many that are uncharacterized. These zinc fingers that just have numbers after them are sort of predicted transcription factors. And it's amazing that these are used by every cell in the body, but their function undiscovered. Many interesting future PhD theses to be developed from that, I think. Okay, so that's one story, one way we're using this atlas to understand how different cell types are arising from the genome. Let me talk about another aspect of human biology, which has to do with aging, and something called the Hayflick limit. So Leonard Hayflick discovered that ordinary cells are not immortal. They can't divide forever. They only divide a certain number of times and they stop. And this was like a revolutionary discovery, because the prevailing dogma at the time of all the big shots in the field was they were immortal. And there were all these experiments showing that cells were immortal, they could be made to divide and culture forever. And it turned out those experiments were wrong. There were artifacts because contaminants, things like that. And Hayflick did the first really careful experiments and showed that, in fact, they'll only divide about 50 times and they stop. And that state of arrest he called senescence. And we now know that's associated with aging. It's been very well studied in the intervening time. It's been sort of one of the key factors. Now you can, in understanding aging, you can take this observation that cells only divide a certain number of times and using the time it takes for a cell divide, do a calculation of the theoretical maximum human lifespan. If you do that, it gets out to be 120 years, which is not too far off from what's been observed. And so the Hayflick limit sort of predicts our maximum theoretical lifespan. Hayflick himself died a couple years ago. And someone at the New York Times had a lot of fun with his obituary. You can see the title, Leonard Hayflick, who discovered why no one lives forever, dies himself at 96. He made it to a ripe old age, which is terrific. So we began to look for senescent cells in our Tabula Sapiens data set. And for those who are into this, we were using P16 markers, cells that are P16 positive, MPK67 negative. So we're displaying the most famous marker for senescence, but not dividing anymore. And through the data set, we found there's roughly 50,000 senescent cells there. They're occurring at about the 3–4% level in tissues, which is consistent with what people thought. And we realized this is maybe the largest, definitely the largest data set of naturally occurring senescent cells anyone's had to work with. And we had it across many different tissues. So we began to dig into that. And Madhav Mantri, a postdoc in the lab, began asking the question, are all senescent cells senescent in the same way? Is there a shared gene expression program that all senescent cells have? And so he began to look at differences in gene expression between cells of the same type, ones that were senescent, ones that were not. And lo and behold, there is no universal gene expression program. There's no single thing that's shared across all of them except in the circular logic, the one that we use to define the senescent cells. And this was sort of really surprising. And these are the genes that are most commonly found and shared among the most, sort of the ones that are most frequently found to be shared between cell types. And, you know, many suspects, many well-known genes associated with senescence were found there, but some that are not and are interesting. And, you know, I sort of earlier in the talk was trying to express the importance of working with model organisms as ways to understand biology. I will now argue the other side, which is that for some things, you really need to look at the human biology. One of the genes here, IL-32, which is one of the most prominent ones involved in multiple cell types for senescence, has no homolog in mouse. There's no comparable gene in mouse. You won't find it studying a model organism. You have to study humans to understand its role. So all is not lost. Madhav did what's called non-negative matrix factorization to look for shared programs and found you know, a dozen or so shared programs, and different cell types we're choosing from those programs, and we can sort of rationalize. It's not that every cell type is senescent in a different way. It's that there's, you know, maybe a dozen or so different ways that they choose different paths of doing that. And you can sort of see that here, where they're sort of mapped out on the top by cell type, on the bottom by tissue. See, there's not tissue-specific effects, but they're about cell types that are shared across tissues, and we can see the same effects in these cell types in different tissues. Okay, so with that, I hope I've sort of convinced you that these atlases as phenotypic companions to the genome are useful and are going to provide very interesting insights into biology. I haven't talked at all about, you know, cell type defined by transcriptome versus cell type defined by physiology. There's a whole nother talk about that I could give. Rest assured, it does seem to correspond pretty well. But I want to spend the rest of the time talking about how we are trying to compute on all this to really get to the, you know, the root of the talk of being able to really analyze all the data at the same time in a way that makes sense. And so while we were building the whole organism atlases at the BioHub, Corey Bargman had set up a bunch of grant programs at CZI to fund groups to do individual tissues, and there were half a dozen or so tissues people were drilling in on. And as part of that, they put together a database of all the data of the people they'd funded, and then they started pulling more data from the literature, and CZI assembled what was sort of a central repository of all the single-cell human transcriptome data that one could find out there. Initially, it was 33 million cells. Now it's up above 100 million, and in the next couple of years, they'll get above a billion. And it was unclear how to use that data. People just thought it would be a good thing to put it together, and conceptually, there was a challenge about how you'd assemble it into a single atlas. It was just a giant matrix of cells by genes. That's why it's called cell by gene. But the jigsaw puzzle wasn't assembled in the way that in a genome is assembled. A genome, you got lots of sequence reads. You would have to put the jigsaw puzzle together to create one whole thing. And so one of the outstanding problems in the field was, how do you assemble an atlas? And I became interested in trying to use AI-based approaches to do that and building large language models on the data. And together with Yuri Leskovich and two very talented graduate students, we decided to take a whack at that. And this is something where we tried to use an approach very similar to the way that ChatGPT or Claude are trained, where we would take this giant matrix of cells by genes and their expression levels and build a neural network that would predict the entries in the matrix. We would cover up 20% of it and then try to find coefficients for the neural net that would do the best possible predictions of the ones that were covered up from the other data. Same way that text prediction is done. Yuri had a terrific idea, which was to do this in a way that was independent of the underlying genome. So it wouldn't be just for human, wouldn't be just for mouse. We would do this in such a way that any organism could be put through this, this model. And he did that using protein language models. So there's I think one of the great innovations in applying these techniques to biology was the work of Rob Fergus and Alex Reeves here at NYU, right in New York, where they built ESM and ESM2, which is a protein language model built on 250 million protein sequences. And they found an embedding space that lets you take any gene and put it in a shared space. And so we use that model here. Every gene was not mapped back to the genome it came from. We put it into ESM2 and used the embedding space value there. So all of a sudden, the genes were not referenced to a genome anymore, but to the protein language model space. And that meant we could compare apples to oranges, essentially, compare cell types across cell atlas data from different species. And so the first model, which we called the universal cell embedding, referees hate that, so we may have to change the name. It was a 600 million parameter model trained on 30-odd million cells over the course of a month. And it looks sort of like this. So if we then sort of re-embed the data after the model has been trained, you can see it colored by cell type, by data set, by tissue, or by species here. And we trained it on, I think, 8 different species. The vast majority was human. So the blue is human, the orange is mouse, and then there's a little bit from lemur, fish, pig, macaque, and frog. So we didn't have a particularly balanced training data set, but it's going to allow us to prove the principle. And so we wanted to test how well this model worked, and had a very strong desire that it did not have to be fine-tuned for different tasks. And the whole goal is, in the parlance of the field, do it as a zero-shot experiment. And so the first thing we did was see what happened when we showed the model new human data it had never seen before. So we had withheld from the training the second Tabula Sapiens data set. It wasn't published, it had never been seen, there's no way to have data leakage. And sure enough, that, shown in orange, mapped exactly where it's supposed to compare to the first Tabula Sapiens shown in blue. All the cell types go to the right places with their partners, with completely different donors and different data. So that was something that made us feel good. Then we decided to say, how does the model do with an entire new organism, a species it's never seen before? So we used a data set from the literature on green monkey, which was not part of the training, and from the lymph node, embedded it in the space and asked what were the nearest human cell types to that. And so if you ask about the green monkey cell type as defined by the authors, whereas what's the nearest thing in the embedding space, it does a pretty good job of figuring out which cell types go where. So this got us to believe that these models would be able to work with species never seen before and do something reasonable with them. And then we began to sort of look more carefully at distances within the embedding space and build phylogenetic diagrams. And so this is sort of, I think, one of the really sort of key insights and opportunities of these sorts of models. It lets you measure distance between transcriptomes. We've known for a long time how to measure distances between genes, and that's how people build phylogenetic trees, to look at relationships and how evolution has changed genes and what are the relationships over evolutionary time. But we've never had a way to do that with transcriptomes, and now we do. So this is showing distances of human cells within the embedding space. And what you see are the cells of a similar type, T cells from different tissues and different flavors, all are close together in the space. They're close to other immune cells, whereas muscle cells over here and endothelia over here. And they're sort of, the distances make sense with what we understand about biology, the biology and the lineage of these cell types. And none of that was part of the training. Remember, there was no biology that went in the model. It was just trained on a matrix of numbers. We did a more general version of this calculation by using the sort of cell ontology tree and looked at average distance between parent-child relationships, siblings, cousins, and more distant relations. And the histogram of distances moves further and further away. So in the most general sense, these distances are reflecting something about lineage. And these sorts of emergent behavior got us very, very excited about these kind of tools. And based on that, we then went and developed a second generation of models at CZI. I started an AI group there, and we called that transcriptomics. And Theo Carletsos led that and began to think about making generative models and ones that are trained more systematically across evolutionary time. And so they built a set of models trained on these 12 organisms, and let me tell you a bit about that. One of them was trained on all 12 of these species, and we called it transcriptomics metazoa. And so it's, I won't go through the whole list, you can read it, but it's spanning just a huge amount of biological distance from vertebrate to invertebrate, and even to single-cell organisms, both yeast and malaria. It covers about 1.5 billion years of evolutionary history. We built another model that was trained only on the model organisms in human. So human, mouse, fish, fly, and worm. And then we built one trained only on human data. They were all the same architecture so that we could do novel species, we could compare results across them. And we're trying to sort of systematically understand, is it useful to train across large evolutionary distance or not, and what are the properties? So there's various ways to test these models. This one is just cell type classification. Given held out data, in this case, human held out data, how well does classification work? And you can see these were the 3 CZI models. This is the one we did with Yuri. This is F1, which is the sort of statistic. And they're all doing very well, better than other models in the field that have been generated previously. Not a lot better. Everything is pretty close. It's sort of all saturated, but nominally better. The CS people seem to care about that. This one didn't do very well. This is from a company. This one is interesting. This was our sort of control case where we weren't doing any machine learning. We hadn't built a large language model. We just took the transcriptome and for every transcriptome, we took the average of the protein embeddings in the ESM space. And even that does pretty well. So another important lesson in all this is you want to make sure that the machine learning is necessary in giving you performance improvements. And you'll see that more clearly in a couple of other examples going forward. So we then said, okay, where is the model doing well and where doing poorly on classification? So here, every row is a different cell type. Every column is a different model. Red is good, blue is bad. You can see all the models are doing well for the majority of the cell types, but there's a small number down here where there's some variation in performance. And if you zoom in on this region over here, you'll see largely where the models are struggling are on T cells. And that's where all the performance advantage is coming from for transcriptomics and UCE. They're doing better on T cells, which is super interesting. And we're still trying to understand why. T cells, immunology, complicated. I think the ground truth there is hard to ascertain because for decades, the way you got a paper published or a PhD thesis or a tenure was to discover a new T cell subtype. And so the classification by experts is a bit arbitrary. And maybe that's part of the issue. Okay, what else did we do? So looking at different species, okay. So here, I think this was cortex, human and 3 other primates, chimp, rhesus, marmoset. Human was part of the training, the other 3 were not. And if you sort of look at where these things sit, colored by organism, you can see there's a bunch of clouds, and each of them have the 4 species near each other. And when you then look at the clouds and color them by cell type, by the annotated cell type, each big cloud is basically one cell type. So what's happening is the model in this embedding space has figured out, even though that these cells come from different organisms, they're of the same cell type and they're clustered close together. So that made us feel pretty good, like some basic biology is being discovered in this embedding space. You can do that across all kinds of evolutionary distances. This is fish and rabbit, and if you ask, what are the closest cell types between fish and rabbit in the space, it tends to be cells of the same type. It does kind of work out over longer and longer distances. And this is something that allowed us to start thinking about evolutionary questions, perhaps. I had the team do the yeast embedding. So yeast, single cell organ. We want to know, how did multicellularity arise? Where does yeast go? What are the cell types it's closest to? Turns out it's closest to progenitor cells and stem cells. That's sort of interesting and something that we're going to be following up on. You can look at other questions. Where did neurons come from? In this case, we had atlases that weren't part of the training, but we could use. Worm and frog, they have neurons. Sponge does not. There's all kinds of interesting literature about, are there neuronal precursor types in sponge that may have evolved in neurons? Two of the candidates are choanocytes and neuroids. These are sensory cells that have cilia. These are cells that secrete things, maybe first part of the synapse. And then we asked, what's closest in the embedding space to the choanocytes from roundworm and from frog? In both cases, it's neurons. Maybe it's right. These are ciliated cells as well. So this maybe is providing some support to the idea that these were neurons evolved from, something like the choanocytes and sensory cells like that. On the other hand, the neuroids were closer to other kinds of secretory gland cells that have nothing to do with neurons, and maybe that one's a false direction. And so this is how we're going to be using these kind of tools to probe these questions of evolution and where multicellularity came from. With that, I think I'll stop and take questions. I hope to summarize, we have experimentally measured the molecular composition via mRNA of nearly every cell type in human and the major model organisms. I wouldn't say it's complete, I'd say it's good rough drafts. These high-dimensional data sets are very rich in biological information, and I talked about how we've used it to understand transcription factor usage across the entire genome, as well as what happens in senescence. We've been able to use large language models as a way to synthesize all of this data in a single unified space. I've given you some examples of where that's going to be useful. And these models are displaying very interesting emergent behavior that might help us understand things like the evolution of multicellularity. We haven't solved the problem I started the talk with of, do we really understand how many cell types arise from one genome? But I think we have the foundation for doing that. And by combining these sorts of resources with our understanding of transcription factor usage and analysis of regulatory regions, we're within years, 5 years maybe, of really being able to unlock this and have a tool that'll help us predict the cell types of a novel organism whose genome has never been seen before. There are also, you may not be surprised, that there are practical applications of these cell atlases in diagnostics and therapeutics. That's a whole other talk. I won't get into that now, but I'm happy to field any questions on that. Thank you. Yeah.
Are you willing to say a few speculative words on what might be the physical factors that, how does this blob of tissue know that it's going to be a neuron? Some precursor chemicals, maybe not even organic?
So you're asking, how does cell type determination, yeah.
Speculate.
Yeah.
You told us we don't know.
Yeah.
But
So, I mean, we don't know how to map it to the genome. Much of the molecular signaling has been worked out by developmental biologists. They've been doing that for many decades, and there's pieces of, I mean, it is complicated. There's beautiful stories, again, in the model organism. Drosophila is sort of the classic one, a zebrafish, another one. So it's, a whole series of complex molecular events. Those sorts of dynamics are not the sort of thing I'm trying to predict. I mean, my sort of approach on this is, let's try to not have to understand development. Let's try to predict adult from the genome. And that may or may not be the right way to go, but my speculation is it's going to be productive. And it will be able to, from insights like this, be able to do that.
I'm just going to make use of the mic because the microphone. So I'll take a question over here.
Just wondering why you pivoted from physical biology, and are you still thinking along the lines of your famous 2006 illustration in Physics Today with Rob Phillips, who was here about a year ago, who says he's still embracing that idea? Are you still thinking along those lines?
Well, I would say the things I work on now.
Virgin energies.
Yes, no. I mean, Rob and I, we're still very close, and we talk all the time. And he, in his beautiful work on physical biology, has been carrying that program forward. I think my interests have become more biological, and questions like this, understanding how to interpret the genome, and I would say more computationally oriented than first principle physics oriented. And Rob and I have very spirited debates about this, but we have converged on a shared interest in trying to understand, fully the genome, and we've been trying to collaborate on a project on E. coli to understand the regulatory structure. Even that, not fully known. And so we're finding ways to weave it in and out still, and we're meeting each other in the middle, I would say. We're meeting each other in the middle. Maybe that's the fairest way to say it.
Here's a question you probably get quite often over here. So on the issue of senescence, and you're finding that there isn't a single program that controls that, does that imply that it will be more difficult to override that annoying feature?
Yes, I think it will. If you're looking for a single magic bullet, a senolytic drug, I think that's very unlikely to happen. Sorry. Make the best of your 120 years is all I can say.
On that same note, could you talk a little bit about epigenetics? What you're learning about that?
Yeah, that's a great question. So we've not been measuring that directly, and there's questions about measuring chromatin state in these cell types, understanding how to connect that to gene expression, and there's been beautiful work. Probably the most beautiful work there has been Stein Ertz, I think, in some ways. He's interested in very similar ideas, and done great things in Fly around that, and I'm thinking that we need to go and start to measure chromatin state in all of our cell types as well. We built a biobank of everything, so with every new measurement technique, we can go back and measure again. And I'm becoming more and more convinced it's going to be important to have the chromatin state as well to really solve the problem.
Yeah, thanks for a great talk. I was wondering about inter-individual kind of variability. Obviously, this is kind of a huge factor, even in, I guess, the original Human Genome Project. Mm-hmm. My question is sort of, in your experience building this kind of cellular atlas, where I would imagine this is a much sort of bigger sort of factor, can you comment a little bit on, in your experience, sort of, what have you found about inter-individual variability? Is it coordinated across cell types? Are you seeing things that are kind of very specific to one individual that you're not seeing in others? And sort of in your estimation, are we close to having kind of this comprehensive like phenotypic manifold of all of human cell type diversity, or do we still have a ways to go yet?
Yeah. Well, we see substantial and very interesting differences between men and women. And, that's been, sort of a very interesting thread to pull. And we don't have a huge number of donors, right? So we're not looking at polymorphism differences, things like that. That's not the question we're trying to solve here. But between men and women, yeah. And, there's an interesting literature on expression differences done on bulk studies. Many people find many, but very small effect. And here, as we do at the single cell level, we see many, and in some cases, very large effects. And that's been interesting. We're preparing a manuscript on that. As far as the question, I think you were really asking about polymorphisms and how that's affecting cell type. Theo Karaletzos has a second paper on that. And I wasn't so involved in that, but I think it's up on bioRxiv, and they think they can predict gene expression differences at the single cell type level based on polymorphisms using the next version of model.
Any other questions?
There's some in the back there. Yeah.
Get on the front first.
Okay, thank you very much. Are there other fields that are accessing your database, like people involved in morphogenesis or anything like that, or expressed interest in accessing your data?
Yeah, well, there's pharmaceutical companies that download it every month, the entire cell by gene. I was down at the ASCB, the American Cell Biology meeting, a year or two ago, and giving a talk, asked for a show of hands, how many people are using it, and it was not as many as I would have liked. It was maybe a quarter of the people there. So, there's folks who are engaged in it and folks who are not, and, over time, I think it'll improve.
Okay. Hi, I'm not a biologist, so my question could be very naive. So it seems like all humans share the same genome.
On average.
On average.
99.9% the same. We're all 99.9% the same.
But frogs also share very similar genome. But how comes the switch? It's what nature is developing, genome, and then individual species. Or first some kind of species come.
You're asking about how speciation happens.
Genome is kind of developing. What is the first? Some individual organism, or first like a code, a genome, and then already individuals?
Yeah, well, that's a very deep and profound question. And you're touching on what's the origin of life and the first cell.
Yes.
And, there's not a lot in the fossil record, let's put it that way, but there's been a lot of speculation and hypotheses, and, many sort of stories people can tell about how that might have happened. And how did the first code arise? And was RNA the first molecule? Because RNA turns out to be catalytic as well as having a code. And, I'm interested in how multicellular things happen, not how the first thing happened. But, the chemists have a lot of interesting theories about the first cell and what came first. And there's many interesting books on that. I'm not sure we'll ever know, though.
Thanks for a great talk. So I'm curious about the senescence example that you were talking about. Mm-hmm. Okay, so you saw that there were a lot of different programs for senescence. And you also saw, or folks saw, that this, I don't remember the right name for this, but when you connect two mice of different ages.
Parabiosis, yeah, it means living together.
Yeah, so there's some kind of, is there some kind of reverse senescence that's happening, and is that the same process, or how does that map to the range of processes that you actually saw? Yeah, so,
what do people measure with parabiosis? I mean, initially it was, sort of physiological effects, and, senescence is not the thing that's fully been nailed down there. We've looked at gene expression differences, so we look at what gene expression changes you see with age. We measured the ones that happen with natural aging, and then we measured the ones that happen with parabiosis and asked, do they correspond to each other? And we can do it both ways, because with parabiosis you have accelerated aging and you have reverse aging. So you can just change the sign and ask, is that consistent? And if you ask what's consistent between all three of those things, forward, reverse, and natural aging, basically the long story short is genes that are downregulated with age tend to be consistent across all that. Genes that are upregulated with age tend to be artifact and are not consistent. That's kind of the bottom line take home of that and how, we think about it.
Hi, my question was about the virtual cell challenge. I don't know if maybe you're already very familiar with it, but I'm not. I didn't actually do it myself, but I have friends who tried it. But I think they were looking for the effect of genetic changes on the transcriptome. I'm wondering if any of these models would be useful, or if it's like the wrong direction to go, transcriptome to embedding space, or any information and connection you have at all. Thoughts about this?
No, I think the competition is well thought out. I mean, the thing to think about is what it means to win. It doesn't matter to have just a very small incremental advantage over a different model. And I was getting at that a little bit when I was showing the cell type class, where everything is saturated in performance. Is it meaningful to have small differences? The machine learning guys love that. And, for things like Netflix recommendations and serving ads, small differences can probably translate into a lot of dollars. But for basic science, understanding biology, I'm not sure that small differences in performance are going to be that meaningful. And that's sort of yet to be probably fully played out.